Record now. My name is Serena Newhall. I'm the HR manager for the University of Washington's Department of Genome Sciences. And today we have Dr. Lee Hartwell with us. Dr. Hartwell is an emeritus professor in genome sciences, was the former director of the Fred Hutchinson Cancer Research Center, and also the 2001 Nobel laureate for medicine and physiology. So thank you very much for joining us. I'm not going to go too much into his career because there's a lot of it available on the internet. And today we're going to ask a bunch of questions that are more related to his life and how things were for him in his career, and some of the elements that one might not normally get to hear about in professional settings, or as well as some questions about advice for those who are graduate students and early career folks. And that said, later on, we will have some space for others to ask their own questions. And so we'll go ahead and begin. So Lee, what were your early years like? Was there a particular event that made you want to be a scientist?
It was a late insight, I think, that arose primarily out of the fact that I was recruited at Caltech out of my first year of college at a community college. And it was really, I think, getting into the Caltech environment that made me realize what science was and what it was all about. Looking back now, I realize that I was always very interested in how things worked and frustrated when I couldn't find out. So I think there was a natural fit, but I didn't even know really what science was until I got into the Caltech environment.
Thanks for that. So how did you decide to study yeast? Celeste Berg said there's an interesting story there.
Yeah, that was a fortunate discovery and decision. I had left graduate school where I studied gene regulation, deciding to study cell division because it was a vast unknown area and seemed like an appropriate thing to do, to study. It was central to biology. I spent about a year and a half as a postdoctoral fellow in Renato Del Becco's lab in Salk Institute and studied animal cells in culture and animal viruses, tumor viruses and things. And I came out of that with the conclusion that it wasn't for me, that I couldn't really do much with tissue culture cells, and was frustrated. I took a position at the University of California, Irvine, but really didn't know what I was going to do. And I'd gotten a grant and I ordered equipment and stuff to set up a tissue culture lab. But in that interval between when I got there and the equipment started arriving, I had some time to think, which is always a useful thing and sometimes rare. I remember Delbruck used to tell people they have to stay away from the lab one day a week so they have some time to think. But that was an undergraduate experience. But at UC Irvine, I started thinking about, what was it that really led to insights in biology? And the two areas that I was particularly impressed with were gene regulation and where the lac operon had been worked out, and also bacteriophage morphogenesis, which I had the good fortune of being an undergraduate working with Bob Edgar in Delbruck's group, who did a lot of that work. And in both cases, it was genetics that was the key methodological approach that opened up those fields. And so I decided that I wanted to do genetics on cell division, and that led to looking just through the literature in the library for a eukaryotic cell that you could do genetics with. And I discovered yeast, which was well developed as a genetic organism, but it was a relatively little known field because it was just formal recombination genetics that people had been studying. Now you're muted, Serena.
Thank you. Thank you. So that was really interesting. Thanks. And it seems like you've researched a number of different things over the course of your career. How do you go from one topic to the next? How do you select which area of research you're going to go towards or away from?
Oh, RJ has like one less tall chair.
Well, I think my own experience.
Oh yeah. Well, we're using the tall chair, like
Joe, could you mute yourself, please?
Cindy and Leah are using the tall chairs for the short chairs. Did you check over on the other side?
Yeah, I'm just gonna go stand.
A little competition there. My sort of peripatetic career has been one where I just really follow what I'm interested in, and cell division was something for quite a while, and that sort of led to interest in the accuracy of cell division. Yeast was a great organism to study that in, and what contributes to accuracy. And the realization that cancer was a disease of inaccuracy, that cancer cells make hundreds to 1,000 times the error rate in cell division, and that seemed like a fundamental difference. And so so that's sort of how the interest in cancer came about. And then the interest in cancer really led to an interest in medicine in general, and an appreciation of the fact that medical science really is a combination of basic science, clinical science, and public health science. And I found the invitation to join the Fred Hutchinson Cancer Center very attractive from that perspective, because it consisted of those three divisions. And at the time I joined, they were all coming together. They were been physically separated. They were all coming together on the same campus. And so it just seemed like a great opportunity to try to learn about how these different disciplines collaborate to, create medical science. That was just the big idea that attracted me to that.
It sounds like a really fascinating combination, and it's neat that that continues to this day. I'm wondering on a different topic, what hobbies you might have, or what did you enjoy doing outside of work, either pre-COVID or currently?
Well, I think it was just mostly raising a family. I had 3 kids, and still have 3 kids, and now a number of grandchildren and 1 great-grandchild, or actually 2 great-grandchildren. So I think between family and science, there wasn't room for much else.
That makes sense. I think a lot of people struggle with that combination of raising a family and having a career. Do you have any advice for people who are in that situation about maybe ways to balance those, or any tips?
Don't travel. Travel is a tremendous waste of time.
Seems like there are a lot more online meetings and conferences these days. Maybe that will help people.
I think it's just great. This ability to meet virtually like this and teach virtually, which I do now, is tremendous advance. I'll spend an hour with you, and if I'd had to travel across the country for a visit, I would have spent 3 days, right?
Right. Travel takes up so much time.
Yeah.
Okay. So the next question I have is who were the most influential people in your life and why?
Gee, there's so many. It's just difficult to sort of just continuous. I mean, I think the most influential person probably was Bob Edgar in Delbruck's group, who I thought did just a brilliant piece of work in understanding how viruses get put together. And that was the inspiration for my science career, really. So, that, but all along the way, I think there have been so many people in the genetics department. I mean, really, just about everybody who was there was an inspiration in some way. Herschel Roman was a great leader and challenger. He would constantly challenge you in one way or another. Walt Fangman was a great colleague, and Brett Byers and Ben Hall, and all of them studying yeast. So it was just a tremendous community of science there, and yeast biology and research. Stan Gartler was an inspiration in human genetics, and kind of enlarging my view of the world. And Larry Sandler taught me a lot of genetics. I had no genetics as a background. I mean, when I decided to work with yeast, I didn't know. I had never had any kind of genetics research or anything, and I'd never worked with yeast. And so the department itself was a real learning experience for me.
I can imagine. I read somewhere that you thought you would start out studying physics. So this was a change.
Yeah, that change was at Caltech as an undergraduate, and I entered as a physics major, but I took a course in biology for just elective, and James Bonner, who was a botanist, taught that course, and it just, day after day, the only thing I can remember is him standing down in the front of the room, waving his arms about how great DNA was. And of course, this was, just a few years after the structure of DNA was discovered. I mean, this was in the late 50s. And it made me realize that biology was going to change dramatically as a result of that insight. And that's why I became a biologist.
That's great. Do you have a favorite memory from your career or your life that you'd like to share with us?
Oh, gosh. How many? I don't know. I think one very nice memory is when I first decided to work with yeast, and I was at Irvine. I just got on the phone and called a couple of the prominent people in yeast genetics. One was Bob Mortimer at Berkeley, and another was, of course, Herschel Roman in Seattle. And I just called him up and I said, gee, I want to work with yeast. How do I do it? And Herschel just immediately said, well, you need to come visit. And so I did. I visited Bob Mortimer for a couple of days. I visited Herschel for a couple of days. Herschel sent me home with this, and Don Hawthorne as well. Herschel sent me home with this micro manipulator that you need to use to do yeast genetics, $1,000 instrument. And he just said, here, can you borrow this? So, it was that welcome and in a very grand style. That was a wonderful way to get started.
Definitely. It's really helpful when people are welcomed into an environment. Let's see, what's next? All right, so you've been interested in teaching and mentoring students. How can one become a more effective mentor?
Well, I don't, have any great insights. I had a wonderful time as a thesis advisor for graduate students. I think the department itself had a lot of support and structure that made graduate education a priority. For me, it was just encourage students to come up with their own ideas, and then encourage them to pursue them and think with them, and try and find what works. And typical experience was that it would take a year or two before an idea would materialize into a result. But the experience I remember over and over again was the fact that it was a single result that opened the door, and you would say, okay, now you've got a thesis, work this out. So I think if you just, follow the science process of being curious and asking questions and having the courage to try to get an answer, it eventually works.
Can you tell me more about the relationship between courage, curiosity, and science? What is that process? Can you expand a little?
Well, at least in my experience, curiosity was the driver. I find it a little discouraging with undergraduates now that so many of them are planning their careers, in great detail, right? What do I need on my resume? And how much will this experience add to my marketability? Whereas I never gave a thought to any of that. I had no idea what I was doing ever career-wise. It was always just whatever interested me. And I don't know that that's the smartest way to do a career, but I do know that it's the most fun.
Does sound like fun. I'm looking over here because I have a list of questions. So kind of building on that, what advice would you have for students, postdocs, and others who are in the early years of their career?
Well, I think, I'm not sure that my experience is relevant to the current, situation. I don't know. But I've never been able to do anything but follow my interest. And, right now I'm just totally interested in education, how we transmit information from one mind to another, and how we come to understand things, what that means to understand. And so that's what I do. But I'm in a very fortunate position where, because of the Nobel Prize, I can have the freedom to do whatever I want. And I don't write grants and I don't publish papers. I just follow my interest. But, that's probably not a viable track for everybody, especially young students.
It does seem like they have a lot of pressure these days to have those experiences on their resumes and have it all planned out.
Yeah, I think so. It's, and I think, institutions who do hiring and promotion and things depend too much on these external metrics of publications and, impact factors and grant sizes and things like that. My experience, of course, in the very early days of biological science and molecular biology stuff was just, trust the people you recruit and try and help them be successful.
Are there any particular qualities that you would look for in someone you wanted to recruit into your research lab or as a trainee? Like if people were not going according to the resume or the CV and all the publications and the book learning, what elements would make someone attractive or make someone you would want to bring into your research lab?
Well, I don't take graduate students or postdocs now. I work entirely with undergraduates, but at any level, it would be motivation. How bad does this person want to learn?
That is a big part of the picture. What was the greatest challenge that you faced or had to overcome?
I don't know. I've been pretty lucky in that things just always seem to work out. I think the scariest thing in my career was when I was president of the Fred Hutchinson Cancer Center and the Seattle Times came out with a front page story on expose of research at the Hutch that was the way they presented it, experimenting on patients irresponsibly was the basic message, and that was pretty scary.
I can imagine. I'm sure that there are lots of human subjects review boards and things and processes to make sure that everything's okay, but I can see how that would be scary to have that in the news.
Yeah, and we ended up taking the cases to court and making our case that way. It was a long process.
That makes sense. Yeah. So what qualities make for a great scientist?
Oh, I think curiosity and persistence. That's what I try to tell the students, just be a bulldog, just be persistent. Don't give up.
There's some students who face additional challenges by being people of color or women. Is there anything that you would recommend to professors or supervisors who interact with new scientists or underrepresented minorities in science? How can we help make science a more welcoming place for folks who along the margins?
Yeah, I've never sort of, this has always puzzled me in one sense, in that I just don't recognize these ethnic or gender differences. They just don't make any difference to me. But I do feel that I went through a big period of feeling an outsider. because no one in my family had ever gone to college, and I had no idea what college was about, or university, or research, or any of those things. And I just sort of, stumbled along and always felt a little bit of an outsider. So I think the biggest thing that one can do is to be accepting. I remember when I was an undergraduate, the Delbrook group used to have a weekly meeting, and Max Delbrook liked to keep the group small. So he would stand at the door and say who could come in and who couldn't. And he would turn professors away and let me in. And a similar experience happened once with Bob Edgar. I was walking by the hallway. He stepped out and said, "Hey, come in, I got an idea." And he explained to me his whole concept of how to understand bacteriophage morphogenesis. And I'm sitting there wondering, why is he bothering with me? I don't have anything to contribute to this. But it was that attitude that we're all in this together. We're all capable and worthy of respect and time. And so I had the good fortune of having that experience early in my career. And I think that's the basic attitude.
That was really well said, and I think a lot of people can take that as an example for bringing people in who might not necessarily be experts in whatever you're thinking about, to help them grow as scientists and colleagues. So what are some of the research topics out there in the world that you think are the most interesting right now? Whether or not you're going to tackle them personally, just what is really fascinating?
Well, for me, I think it's people. We've done pretty well now with clonal organisms where all individuals are the same. And even with some complex phenomena. But people and other outbred organisms all have this intrinsic variability that makes the analysis and the conclusions much more of a statistical nature. And now with the ability to collect so much data and analyze it using sensors, I really think the big frontier, it seems to me, is understanding people better.
People are complex and fascinating.
I'll say.
So you've had a wide range of service at the national level, from the NIH to the NSF, NHGRI, NCI, HHMI. What advice would you have for people who are interested in service at the national level? Let's say that they're not enough faculty or researchers and they're not there yet, but they might be interested in heading in that direction. How did that come about, or how does one do that?
I don't know. I never felt like I was very effective at that level. I think the only time I was when there was a new director of the NCI, and I just knocked on his door and said, "We need a proteomics program in cancer." And he said, "Okay, let's do it."
That's great.
I helped create that. But I don't know. I always found it frustrating on these big committees and trying to judge grants and various kinds of things. I don't feel like I was very effective or fulfilled at that level.
I guess it's hard to deal with some of the politics and processes, and especially when you have a large committee of folks, it's hard to get consensus or move
Well, I think
forward.
it takes a different mentality. I mean, I think there are people who are much more community aware than I am, and concerned with community issues that need to be dealt with at that level. And my mind just seems to gravitate to individual problems that I want to think about.
That makes sense. It takes all types, and you've certainly had a lot of really fascinating, interesting problems that you've been looking at. So you were head of the Hutch for a long time. What do you think is the most important quality for being the head of an institution?
Probably do more for the institution, not to be head of the institution. Again, you know, I'm not sure I was very effective at that, but it was a unique period when, as I say, you know, we built the campus all came together, divisions all came together. We did a lot of recruiting, a lot of hiring, but the basic culture of the institution is what's important. And culture is a long-term thing. And I think maintaining the good qualities of the culture are probably the most important thing. So the culture at the Hutch had been built largely by Bob Day and the different division leaders, particularly Don Thomas in the clinical division. And I think respecting that culture and trying to maintain it is an important issue.
Okay, this is my last question, and then I'll open it up for others who might have questions or let you speak freely on a topic of your choice. But what do you think are the highest priority topics in science right now for people to focus on, or yeah?
Well, I would just go back to, you know, trying to understand the complexity of outbred populations and people, you know. That seems to me a great challenge because of the intrinsic complexity of it, and the fact that it's there and we've got to try to deal with it. And there are, you know, it seems to me the advances in science always hinge on technology. And we have new technology now in sensors and ability to deal with large amounts of data, and that seems like the thing I would be wanting to do if I was starting my career.
Yeah, all the big data topics and proteomics and bioinformatics are super hot right now. All right, well, thanks very much for answering those questions. I'd like to open the floor now to anybody who would like to ask a question. You can feel free to just unmute yourself and ask your question.
Hey Lee, it's Raghu. Could you say a little bit more about what you're doing in your teaching initiative at the moment? Like what does that consist of, and who are you teaching?
Yeah, I'm hi Raghu. I'm very interested in this whole thing about learning. I think learning is magic. We don't realize it for the most part. But you know, because most of our learning takes place as young children, and we I'm particularly focused on vision and visual cognition as learning modes. And we learn to draw, to make sense of the world visually very young. And we don't realize as adults anymore what we're doing, but it's extremely sophisticated, complicated, and it's magical because our visual capacity is really very limited. Our whole cognitive capacity is very limited. In our vision, we only see clearly out of the about 5 degrees of our visual field, and the rest of 180 degrees is actually very vague. And we only make sense of our visual field by our eyes jumping around, looking at different things at different times. And it's our poor peripheral vision that's deciding where to look next. And we're doing that in a completely unconscious way. Our ability to attend to something is we can only attend to one thing at a time. That's pretty limiting. We can only hold a few, half a dozen visual objects in our mind for a very brief period of time, about a second. So we have tremendous performance limitations, and yet for learning, and yet we do amazing things in a very complicated visual world. So that's the basic problem. That's so interesting, but that's just the backdrop, because I use that platform because I want to create an authentic research experience for naive undergraduates. And I think it's a shame that many undergraduates don't really understand what science and research are about until they get into graduate school, because we spend most of our time in undergraduate school teaching them things we already know rather than what we don't know. And it's the things we don't know that science spends its time on, and it's a different process. So I got into this vision stuff because I was interested in finding an area where it didn't require a lot of equipment, and it didn't require an extensive amount of knowledge. And so we all have vision and we're all familiar with it. And so this was something I could work with. So what I'm trying to do is trying to create a research program where undergraduates are doing the research, and the questions we're trying to ask have to do with how we learn. And would you see where it goes? I just don't know where it's going to go. I don't have any answers. I have lots of questions.
Actually, that sounds like real fun.
It is a lot of fun.
Lee, did you say you were doing this at UW?
Hi, Jerry.
Hi.
No, I'm doing it at Arizona State University,
But you're in Seattle.
but I'm in Seattle because I can do it all online.
Yeah. So have you been in the same room as the students you're working with?
Oh, I have been. Yeah, it started out as, in-person stuff, but as the project has progressed, we do our experiments now all online.
Okay.
And so I think, there's no real need for the in-person environment. We don't use a research laboratory, we just use our computers.
Yes. I would agree with that, but don't you think that personal interaction of being in the same room for some time is important?
Very likely, yeah. Sure, I do. And what I do think is very important is an authentic interaction.
Yes.
And so I have very small classes. I have two classes I'm teaching now, each section has just 5 students in it. And so we have quite a bit of sort of face-to-face time, at least.
Yeah. Certainly Zoom has made this remarkably possible since COVID hit. But my own experience is that personal interaction is built up most deeply and directly by being in the same room or wherever with the person, and that's important to establish a connection.
Yeah, I'm sure you're right about that.
Hey Lee, this is Stan Fields. Great to
Hi,
see
Stan.
you again. I wonder, are there some topics and interests of yours that might intersect with genome sciences so that we could have more interactions with you in the future? It would be wonderful if we could bring you back into the department in some ways that you would be interested to do.
Well, I'd enjoy that. So I'd love to find that venue. I think the idea of creating a research environment for undergraduates where they're really doing authentic research is useful, sort of unique approach. And I could certainly think about, doing that at UW. How it, whether it's genomic or not, I don't know. That's another question.
I think for
It doesn't matter to me, what discipline it's related to.
Yeah, I think for undergrads doing research, they might not care too much, even if there's some remote genetic component, that would probably be plenty.
Well, the component that is there is that we create experiments. Students create experiments on measuring different kinds of cognitive performance, and what you always find is, of course, is that there's individual variation. And so studying individual variation in all kinds of different, any test that you create for humans to perform in, you will find in reproducible individual variation. And then another question that always arises then is, can you learn to perform better by practicing? And then a question after that becomes, can you achieve automaticity? Automaticity is where you no longer need to pay attention and you can still perform well, like riding a bike, or driving a car. So I guess I'm getting carried away here, but there's I think there's a lot of fun things to do with undergraduates in this field where we're very familiar with our vision, but it turns out to be a great mystery once you start looking at it in any detail.
Hi, Lee. Clem here. How are you?
Clem. Yeah.
Good to
how are
see you.
Yeah.
Your comment about learning visually at a very young age reminds me of an experience that I had at Cornell as a postdoc. We were visiting a young couple with an 18-month-old child who was sitting in his diaper reading flashcards. And it boggled my mind. So we went home and made up a bunch of flashcards for son Jeff. And by age 2, he was reading about the same, about 50 flashcards. And F. Racker was visiting at the time, and he made out a card with F on it and was very pleased that Jeff could read F in it there right away. And his young friend at Cornell continued his young learning, and by age 3, he was able to read recipes and bake cookies. So my feeling from all of that was that the potential of young children is tremendously underestimated, really underestimated. I don't know, what are your thoughts?
Well, I think it's under-realized. I think that what children are learning is profound as young children from their visual world, how to make sense of their visual world. They're not under-challenged. They are learning tremendously. They can learn other things. And, you know, for example, now kids are learning, you know, how to use, you know, phones and things at very, very young ages. So we're certainly entering a new era with regard to the kinds of visual information that children are going to learn to deal with.
I just have the feeling that younger children, very young children, are probably capable of far more than we challenge them with if presented properly.
Yeah, I have a little look at it in a little different way. I think we don't realize how much they're being challenged to just cope with a very complicated world they're in. I think they're actually doing very complicated stuff. They are learning very complicated stuff.
Well, I'd like to thank you also for connecting me with Jerry Van Slyke, who designed a house for us that we ultimately built on Bainbridge, a Poland beam octagon, 3 stories, which was an incredible challenge, but it was really wonderful working with him.
Yeah, he was a great guy. Well, it's been so much fun visiting with you. I see Joe Felsenstein, and good to see so many of you are still there. I was so really surprised when I, you know, after getting this invitation, I stopped to think about, you know, well, let's see, how long has it been since I was in the genetics department? And it has been 25 years. I just couldn't believe it.
Lee, can I ask you a question? What are you learning these days?
I think what I'm trying to learn is something about learning in the sense of how we help one another learn, how the social aspect of learning, of communicating from one mind to another. That's what really interests me, and how we represent information in a way that someone else can understand it. I think we have a number of sensory modes through which we take in information, but the primary one is visual for humans. And so it's really fascinating just to think about that. I don't know that I will be able to make any contributions to it, but it's certainly loads of fun to think about.
What led you to this pursuit?
It was the original goal was to create a research experience for undergraduates.
I see.
And I didn't want to have an expensive laboratory, and I didn't want them to have to learn a lot before they could do it. And so that led me to sensory perception, and we did a variety of different, I've done this for a few years now, and before settling on vision as a focal point.
It looks like Carrie Boak has a question. Carrie, would you like to ask your question?
Sure. I have kind of a half-baked question because I was intrigued by your use of the word cope versus kind of challenge. And I have school-age children, and maybe you're aware that in Washington State, as well as I'm sure lots of places around the country, during COVID, test scores have gone down quite a bit. And I suspect that this is because kids have a lot to cope with now. So the kind of, there's more kind of crap that the kids have to deal with nowadays, and it's affecting their kind of ability to learn. And, kind of historically, and still I'm sure it's exaggerated now, some kids have to cope with more than other kids for various demographic reasons, right? So I'm wondering if your research is are you actually looking at this coping aspect of and how it interacts with learning? Thanks.
No, I'm not directly, but I am very interested in my ultimate interest is roughly 7th grade students. Because I think that's when kids sort of decide whether they like science or not. And from then on, it's often too late to help them understand how science illuminates the world. And from that perspective, I'm very interested in science teachers because they're the ones that do it. I think science teachers often get very poor education. There is now, about 10 years ago, the National Academy of Sciences and National Academy of Engineering and National Research Council and stuff came up with this next generation science standards. And the next generation science standards encourages teachers to teach much more from an inquiry point of view, encouraging kids to ask questions and have discussions and things. And teachers don't, for the most part, know how to do that because that's not how they learned. They think they have to have the answers. And questions are much more interesting than answers, and questions are much more motivating. So I'm really interested in how we can make that transition to a more inquiry-based education at the grammar, K through 12 level. And I do a lot of teacher workshops online too, with various places in the world. But it's an international problem of I think trying to change what we ask students to do. So, the standard tests that we're looking at are just measuring the wrong thing, as they often are. It's not getting the right answers so much on a sort of algorithmic approaches that we want kids to do, but we want kids to be curious enough that they want to learn themselves. It's the motivation and the curiosity and the persistence that makes for learning.
Lee, this is Joe Felsenstein. Hi, great to hear from you. I've been particularly concerned by since I do work with mathematics in biology, with how panicky students are about math. And it seems to me it's actually gotten worse. It seems to me it's hard to separate that. It's hard to tell because maybe we, in the previous days, concentrated on the students who could handle it better and ignored the others more. But do you find when you're doing your work on the issues of vision that the students are having difficulty thinking in any mathematical way?
Well, much broader than my own experience. There has been a number of studies, and one very good one that took a huge survey of biology teachers and asked, what are the core needs for education? And the top 6 items were all had to do with math. There is. So I do think mathematical thinking is the crux of helping students gain confidence in their own abilities in any kind of science. I actually meet weekly with a group of mathematicians from Michigan University, and we talk about these issues every week. It's been going on for a couple of years. But
Well, I was just thinking that it isn't even a matter of whether people understand formal mathematics. It's that, you know, they should be encouraged to just become friends with numbers and compute, measure and count things, and find that that's a useful way of thinking. And I think we must be failing way down at that early level.
yeah, I agree. And there are some really nice materials available now. There's there's 12 books available online free called The Art of Doing Mathematics, put out by Berkeley, which is all inquiry based and very appropriate for young students. And then there's another great website where this guy, I forget his name now, has developed a whole series of approaches to math based much more on vision, visualizing relationships between things. But yeah, thanks for bringing that up, Joe. I do think that mathematical, quantitative thinking, relationship thinking, formal relationship thinking between things is not emphasized enough at those young early levels.
Thanks. We're just about at time. I'd like to thank you, Lee, for joining us and answering all of our many questions. It was lovely to have you today. And yeah, thanks everyone who joined and asked a question.
Thank you. It was a real pleasure to be with you and see old friends.
Wonderful. And if anybody's interested in sharing their own life story, just shoot me an email, serenn2@uw.edu. Everyone is welcome to tell the story of their life and how they got here, what their interests are. We've had people on this program from undergrads to grad students, postdocs, staff, and faculty. So I hope that there are others who will volunteer to share with us about their lives in the future. All for now. Thank you.
Bye.